Okay, everybody, so here is the video of the Chapter 5 and 6 PowerPoint. I combined these two chapters into one PowerPoint because they are very closely related. And, I mean, it's good to split them up into two chapters, but I just figured we'll just keep it simple and keep them together. Okay, so the first topic is metabolism. That's what Chapter 5 is all about. Now, you should have learned a little bit about metabolism in A&P 1, and then in A&P 2, you actually learn a lot more about metabolism. But of course, that's everything about humans. So of course, now we have to talk about the metabolism of bacteria, which is in one way a lot simpler, but in another way it's a lot more diverse because human cells can only do so much, whereas bacteria have evolved to be able to do some really incredible things that human cells would never be able to do. So hopefully you already know the terms catabolism and anabolism. So catabolism is the breaking down of materials. Anabolism is the building up of materials. So you break down polymers into monomers, and then you build up polymers from monomers, okay? So when you combine all of the catabolic and all of the anabolic reactions, that's what metabolism is, the sum total of all chemical reactions within a living organism. So a human, a dog, a mushroom, a bacteria, okay? Every living thing has some sort of metabolism, some sort of way in which they use, store energy, obtain some sort of food material, all that kind of stuff. All right, so hopefully that's just a review from A&P 1. Something else you should have learned about in A&P 1 are enzymes. So enzymes are specialized proteins that speed up chemical reactions. So a lot of times you'll hear the term catalyst when we describe enzymes. Enzymes are very important in metabolism because for the most part, enzymes do the breaking down and the building up. Bacteria have enzymes just like humans do. So an enzyme binds to a substrate. So the substrate is the molecule or the material that the enzyme is going to do something to. So the enzyme is either going to break down a larger substrate or it's going to build up smaller substrates into something larger. The place on the enzyme that the substrate binds to is called the active site. Now, when I say enzymes are specific, that means that enzymes only work with a specific substrate. And I'll give you an example about that in a second. Enzymes also operate at optimal conditions. Mostly we're talking about temperature and pH when we talk about optimal conditions. The other cool thing about enzymes is that they are recyclable, which means that once they do their reaction, once they do their job they're supposed to do, they're unchanged, so they can go on and do more work. So here's a picture that I stole from Google a long time ago. Love this picture. It's very, very simple, and it basically shows you everything that I want it to show you. So the enzyme is the gray thing. It kind of looks like a dinosaur footprint, right? And you'll see that the enzyme has the active site. The active site is where the substrate binds to. So when the substrate binds to the active site, and notice the active site shape, the shape is designed for that specific substrate, okay? We call this the enzyme substrate complex, all right? Now, in this case, this enzyme would be a catabolic enzyme because it's taking this substrate and it's breaking it down into two smaller products. So we would say that this enzyme is catabolic, all right? So let me give you an example, all right? You all know what lactose is, okay? Lactose is milk sugar, okay? It's the sugar from milk and milk products, all right? Now, bacteria, for the most part, they really, really like lactose, which is why milk goes bad so quickly because, once you open up your jug of milk and expose it to the air, the bacteria are going to get in there. They're going to start, basically eating the lactose, and their waste materials are usually some sort of acid, and that acid is what causes the milk to spoil. So if this substrate in like the neon green color, if this was the sugar lactose, the name of this enzyme is lactase. Oftentimes, enzymes end in ASE. So if the substrate is lactose, the enzyme is lactase. Lactase is only going to work on lactose. It would not work on other things like sucrose or starch. Those things have their own enzymes. So now over here, if we're breaking down the lactose, then these two products would be glucose and galactose, because glucose and galactose are the two monosaccharides that make up lactose. Okay? So that's enzymes. And in your book, your book has some more updated pictures, so you can certainly look. Pages 112, 113 have information about how enzymes function. Now, some enzymes have what we call a coenzyme or a cofactor. Basically, it's the same thing. What that is it's something that activates the enzyme, okay? Sometimes cells will make a inactive form of the enzyme so it can just kind of like be ready to go when it needs to go, all right? So then when it's time to actually turn the enzyme on, the cell will release the coenzyme, and the coenzyme is basically what activates the enzyme. It's what turns it on, okay? So what affects the activity of enzymes? And I already mentioned these. Temperature is a big one, all right? So enzymes work at an optimal temperature, which is, for humans, the enzymes that we have in our body, they work the best at 98.6 degrees Fahrenheit or 37 degrees Celsius, right? Because that's our body temperature. So whatever the organism's operating body temperature is, that's what their enzymes like. Okay, most bacteria are happy anywhere between 50 degrees Fahrenheit and say 110 degrees Fahrenheit. So definitely what we would consider room temperature, as well as what we consider to be our body temperature. Okay. Now, one way to affect the activity of enzymes is with heat. So increased temperature causes something called denaturation. Denaturation is when a protein is altered and destroyed, okay? So temperature can do this, as well as various other types of chemicals, radiation, other things, okay? But temperature is a big one. And this is kind of one of the reasons why Pasteur figured out a long time ago that if you were to heat up or boil a substance, you would kill the bacteria. So when you boil water, any bacteria that's in there, you're going to denature their proteins, and that causes them to die. pH is another one, very similar. So, most living things are happy at a neutral pH. That's why we don't have very many enzymes in our stomach, because our stomach has a pH of 2. So there's not many enzymes in there. There's also not much bacteria that can survive that pH, because the pH is very low and very acidic. So one thing that just, it makes so much sense it almost doesn't even need to be said, but the substrate concentration. So the amount of substrate is going to affect the enzyme's efficiency, right? So if we're talking about something like lactase, well, if there's no lactose around, there's nothing for lactase to do. And oftentimes, the cell won't even produce the enzyme unless the substrate is actually present, unless it's actually there. Now, inhibition is a really important topic for us, okay? So inhibition is basically turning the enzyme off, or, blocking the enzyme. That's what inhibition is. Why do we care about this? Well, it's part of our arsenal. It's part of the way that we can fight bacteria. Because if we block or destroy the bacteria's enzymes, we're going to stop the bacteria's metabolism, and we're basically going to shut down the bacteria, so it won't be able to divide anymore, and it won't be able to really make us sick. So there are inhibitors that are called competitive inhibitors, and then there's non-competitive inhibitors. And if you remember, I briefly talked about sulfa drugs in chapter 1. Sulfa drugs are actually competitive inhibitors of bacterial enzymes. And what's great about sulfa drugs is that they do not affect the enzymes of the human. So they only target the bacterial enzymes. And of course, now that we have problems with antibiotic resistance, sulfa drugs were kind of going back to the research and the development that was being done with sulfa drugs, because we need like another way of killing the bacteria besides antibiotics. So this just summarizes the two types of inhibition that I just mentioned. Competitive inhibition is when the inhibitor binds to the active site, and that effectively blocks the active site. So the substrate, so in this example, we have two smaller substrates. So this is going to be an anabolic enzyme. It's going to make something bigger. But these two substrates, they're trying to bind, and then they can't, because this inhibitor is blocking the active site. So they're trying to get in there and they can't. All right, that's competitive. Non-competitive inhibition is when the inhibitor binds to somewhere else besides the active site, and we call that the allosteric site. So this is kind of like a little bit more indirect. It's a little bit sneakier, okay? What happens is the inhibitor is going to bind to the enzyme that causes the active site to change its shape, and now the substrates can no longer bind. So the active site is blocked in a more indirect way, because now the active site has changed and the substrates can no longer bind. Okay? So competitive inhibition binds to the active site. Non-competitive inhibition binds to an allosteric site, which has an effect on the active site. Okay? And we will come back to this picture. I only have about a minute left in the video, so we're going to go ahead and stop it there. Next video, we'll pick up with feedback inhibition.